Genomics refers to the study of the genome. By the genome, we refer to the entire DNA set in a given human being. Half of it is endowed from your biological father and half of it from the mother. When we say genomic testing, we're referring to the use of DNA sequencing technologies to understand the DNA of a patient in a clinical setting. It is very important because we tend to understand what the underlying mutations are of a given disease phenotype. In this age of genomics, it really helps out the doctor to plan out what therapeutic regimens are going to be used for that patient. We need to understand the patient genome in order to learn what mutations the patient already has from a familial perspective, and B, what are the mutations the patient has been accumulating from a somatic perspective. Genomic data also helps us understand the sequences of your metabolic genes. That really unfolds and unravels how your body is going to metabolize a given drug. Again, this goes back and helps the doctor to plan out what doses you require for a given treatment. Having said all of this, genomic technologies are really challenging. For one, it really needs a lot of different sets of talent to make sure the data is understood and the insights taken to the clinic are relevant. It all begins with your doctor. We get a sample, it could be a blood sample or any other sample that is processed. By processing, we mean a biologist takes over and extracts the DNA and prepares the libraries which will be used for the actual reaction in the NGS machine. Once the run is complete, the data scientists take over, the bioinformaticians, and they assemble the entire genome and assemble it with the reference sequences as well to understand what variants occur in your sample. After that, we need a lot of clinical insight on what variants are relevant and what could be ignored at that stage. And it finally ends with genomic reporting, and by that we mean what sort of insights are finally going to be taken to the doctor for him or her to decide what your treatment should look like. So you can clearly see a whole pipeline of different talent being used just to decipher what your sample holds from a DNA perspective. The Center of Genomic Sciences is very lucky for a diversity of reasons. For one, it sits in one of the densest healthcare clusters in the country, the Phase 5 of Hayatabad in Peshawar. We see a lot of patients from different ethnic backgrounds. That gives us unprecedented access to the diversity of DNA and DNA sequence data. Second, we're piloting out the country's first precision medicine pilot. That means we're backed by some of the brightest in the field when it comes to clinicians, biologists, or data scientists. This also means we have all our workflows and our protocols streamlined and fully functional and ready to blaze through any number of samples. Finally, we're not restricted to a single technology. We're scientists to the core, and we love to experiment with different technologies. Whether it's the Illumina machines or the Oxford Nanopore gadgets, we love to dabble into all these areas. And we've developed all the pipelines that we require. As RMI, we have a huge footfall of patients that exposes us to a wide range of rare disorders, which are always challenging, and we welcome all these challenges at the Center for Genomic Sciences.